regulation is a process where the body decides which genes should be expressed. In this example, gene B is not expressed properly in the liver. There are many ways that this could occur, but one way is through DNA methylation. DNA methylation plays a critical role of gene silencing by remodeling the chromatin. Methylation involves the addition of a methyl group to cytosine using the enzyme DNA methyltransferase. Once this methyl group is attached, it can cause a physical impediment of transcriptional proteins to the gene. In other words, RNA polymerase may not be able to reach the promoter due to the large methyl group. The more cytosine groups in the DNA will bring about more CpG-rich areas, which promote methylation. And CpG just means cytosine, a phosphate, and guanine. And in mammals, about 70 to 80% of CpG cytosines are methylated. Methylated sequences can easily acquire histone deacetylases, which further modify histones into condensed heterochromatin and become ultimately silenced. Consequently, an abnormal amount of methylation in the liver would cause this gene B from not being expressed. However, if this is not the reason and the eukaryote gene is transcribed as normal, there are still ways this pre-mRNA molecule may be altered at gene B. One way this occurs is when a pre-mRNA folds over itself to form a microRNA. There are specific microRNA that must bind to the mRNA in the liver to regulate gene expression. The microRNA will bind to the mRNA through base pairs and completely inhibit the mRNA from being translated. In this case, there are a high number of microRNAs produced with large expression levels to silence gene B.